All right, everybody, hello and welcome to the second piece of our experiments lecture, which is more specifically experimental design. So last time we talked about the basic elements of an experiment and that they are the only type of study that can infer cause and effect between two different variables. But we have a few different means of doing so, some more nuances of design that can help us to answer different types of questions. And we'll also be talking about the limits of experimental design here. So let's start with our sort of halfway in between experiments and other types of designs. So if you remember all the way back to our first lecture, we had discussed three major domains of research. We discussed descriptive studies, correlational studies, and experimental studies, each of which have their own set of considerations. Descriptive studies have no manipulation of any variable. Correlations are the same, but we're comparing two variables to each other. Then experiments are more specific in that we actually manipulate or change the independent variable. However, we can't format everything as an experiment. We can't manipulate every variable out there. An easy one to take a look at, for example's sake, is gender, right? We do lots of studies on gender where gender is a predicting variable, but it's not something that we can manipulate. Therefore, we can't have causal studies about gender. We can't say that gender causes anything specific when we're talking about psychological phenomena because we don't have random assignment. We can't randomly assign individuals to two different gender groups or three different gender groups because we can't force it, right? It's not something that we can manipulate. So that's why we call these non-manipulative or quasi-experiments, because we can otherwise format this or structure this as an experiment where we maybe manipulate levels amongst our groups, but we can't actually manipulate those groups themselves. So these are things like correlations. This is what we'll be doing with our surveys. We'll be doing quasi-experiments. We will not be interfering with or manipulating anything to do with our participants, right? We'll just be assessing information that they're willing to give to us. So generally, we have to test more individuals if we're going to go with this type of design as compared to true experimental designs where we have more control of the study. And we'll talk about this a little bit more as we go in the class. Larger samples are generally revered as better because the larger the sample, the closer we get to the population. However, when we have less variables, we can control for the larger the sample is, the more we can hone in on a specific variable. Right, so we're going to talk about one major variable that exists in all studies, no matter how you slice it, and that's the passage of time. We can't control time at this point, but we have to include it or consider how it impacts our studies. And most importantly, time is a variable we consider when we look at things like lifespan development. So a lot of what we're talking about here is age of our participants. So the first type of design here we have is a cross-sectional design. In a cross-sectional design, we take multiple groups of people of different age ranges and examine them at the same time. At the time I'm filming this, we have a few years ago, we just got out of a great example of cross-sectional research, right? COVID. So cross-sectional research will be done if we want like perceptions of children and adolescents and adults on the COVID-19 pandemic, right? We would have to ask each of those groups separately, right? We can't ask one person, say a child, and ask how they would interpret it as an adolescent, right? It doesn't work that way. If we're interested in multiple age groups at a certain point in time, we have to go with this cross-sectional design, pull individuals from each of those age groups and compare them at once at the same time. We have a sort of counterpart to this where we take individuals and we study them over their lifespan. So we talked about control and experimental groups, and in longitudinal studies, you become your own control, right? We're comparing yourself to a baseline of your past self. So longitudinal studies are considered a sort of gold standard in developmental research. However, they're very hard to do, very expensive. It's very hard to get individuals to participate in research to begin with. It's even harder to get someone to agree to participate in your research for their entire life. So for many, many years, they have to commit to this. So we measure the same things across all occasions over the lifespan Some of you may have even been involved in a longitudinal study when you were involved in your K through 12 education. There's a lot of research being done on, say, health variables, tracking the youth in our country. So we have some issues with this with longitudinal studies. If we think about longitudinal studies in just the psychological sphere, when we're recruiting individuals from the population to participate, people will drop out, right? People might move away from where the study is being conducted. And unfortunately, we have even death as an issue in longitudinal studies, mortality. We can lose people to this, especially if we're looking at disorders that generally occur later in life. We have natural maturation changes, so your baselines aren't always going to be completely indicative of your past performance because we do change over time. So this says threats to internal validity here, and that's actually a term that we'll come back to when we talk all about validity in the future topics. It basically refers to the accuracy of a study. There are some threats to accuracy because of the difficulties of conducting this type of study. So here's a wonderful infographic comparing the different types of comparisons we can make. We have cross-sectional comparisons in red, comparing two different age groups at the same time, or two different age groups. I mean to say we have cohort comparisons, not something that we talked about here, but comparing kind of across years to those of the same age. Then we have in blue here longitudinal comparison, so looking at the same individuals as they age longitudinally, right? So I think that this is helpful to get an idea of the different types of comparisons that can be made and what your study would demand, right? Some studies demand that we use cross-sectional research and make these comparisons across different age groups. Sometimes longitudinal or continued research on certain sets of individuals would be preferred. So we're going to talk a little bit about statistics and experimental design. And this is mainly to highlight the limits of experimental design and really of all research. But we'll start by just talking about the p-value. So this is not the first time we've talked about the p-value, but let me remind you that it's a probability. All right, p-values are probabilities, and it's the probability of obtaining a result as extreme as the observed results if the null hypothesis is true. And I know that's a mouthful, so let's give this a little bit of thought, all right? I always default to sleep because I feel like it's easy to understand. So maybe you read some research published by like Cal State, and they say college students in this area sleep on average 9 hours a night. And you say, hold up, all right, I don't think that's true. Maybe you have a hunch about this. Maybe you've done some research on this and you say, hey, I don't think that's true. I think that college students on average are getting less sleep than that. So when you walk into a study that, 9 hours of sleep would be your hypothesis, that's what you're testing against your hypothesis, would be testing something different from that. And in order to determine if your hypothesis is correct, you would need to find something very different from that null. And in fact, so different that there was a less than a certain percentage of chance of it happening. That certain percentage would be our alpha level or our p-value, which we usually set to 0.05 or 5%. All right, so I know conceptually this is a little bit strange, but if we can think back to our time and in statistics, we're looking for when we find statistics in those critical regions, see where it says reject the null, and I have an all famous saying in statistics, when the p is low, the hoe has to go, right? The null hypothesis, or the H subscript 0, often referred to as the hoe of the HOA of the null and alternative hypothesis. When the probability of what you observed is low, that's when you reject the null hypothesis, right? Then you have some evidence to suggest that your hypothesis is true. It's right. We don't say that it's true, but we say that we have some evidence to suggest it's true, right? Maybe you do a survey and you find that college students are getting on average 5 hours a night. We'd have to run a statistical test, but that's pretty different, right? So different that it might be statistically improbable, in which case that null would have to go. So this becomes an issue specifically with experiments in this type of design, because the more instances, the more tests that we have, the more likely we are to see significant results. This sort of goes back to the larger our samples, or the more likely we are to see significant results. It's all a bit related here. However, we often run multiple comparisons in research. We have lots of different tests that we want to make. Maybe we're running correlations between the amount of time you sleep and the amount of time you work, the amount of time you go to school, right? What your diet's like, how often you exercise. The list can go on, and we can start to compare all of these different variables to each other. And we can get multiple comparisons, and what that means is the more comparisons we have, the more likely we are to detect significant effects. And this is just due to probability, right? Just basic probability. If we're testing at an alpha level of 0.05 and we run 100 tests, 5 of them should come back as statistically significant, just due to the probability. So it's a little bit strange, and I think that this particular example is a humorous way to wrap our heads around this idea. The more variables we have, the more comparisons we make, the more likely we are to find a significant effect. So here we have an example with jelly beans. If you test every color of jelly bean and something else, in this case they're testing the link between jelly beans and acne, what if you find one, right? You test 100 different colors and it comes back, hey, green jelly beans are statistically significantly related to acne. What you do with that information? Do you go out and put it on the front page of a newspaper, right, and boldly exclaim that you have found the cause of acne? No, you're not going to do that, right? Because there are more considerations here than, meets the eye. First of all, this could be an error, right? That's what our error rate determines, right? So the family-wise error rate is the chance of finding significant results based on the number of tests or comparisons. If you run 100 tests, you should expect 5 of them to come back significant, just probabilistically, just random chance, right? So you might be thinking, what the heck, then how do we ever know if what we're getting back is correct if we have this percentage rate of error? Well, that's where our literature review comes in. That's where our overview of theory comes in. That's why formulating our hypothesis and writing an introduction is so important before we start to go test things without any sort of backing theory. So we always need theory to back up our statements. So when we find something like this that might have just been a coincidence, we don't report it as the truth, right? So I like here on this paper, it says only 5% chance of coincidence. But remember, 5% can happen. It's unlikely, but it's possible. It's not as unlikely as you might think. So that's why statistics aren't our wonderful blanket answer to everything, to all questions. We always have to consider the limits, right? The rate of error, and how our findings might not be fully representative of what's really going on. This is why the scientific method is also a circle, right? A cycle, so that we can repeat this again and again. If we did 10 more studies and green jelly beans are the only ones that come back significant every time, then you might be onto something. But one case is not enough to buy into that theory, right? So in summary, we have, experimental research. We have some issues with it, right? Some issues with experimental research. One that we haven't touched on so far here is artificiality, or the artificialness of experiments. If any of you have taken part in a true experiment, you might know that you're often taken into like a laboratory setting, in an unfamiliar setting, and because of the idea that we want to strip away any variables except for the independent variable, we often put people into somewhat unnatural circumstances. And we might not be getting an accurate read on their behavior because of this, right? And another issue with experimental research is our ability to manipulate variables, as we discussed with our quasi-experiments and non-manipulative studies. Some simply cannot be manipulated. Right? We can't manipulate someone's level of education for our study, nor could we manipulate gender or their socioeconomic status or their type of parenting, whatever it may be. We can also only infer the cause of the independent variable. So remember, cause is a bit of a heavy word for us. Here we can't apply cause to just anything. And in this case, we can only apply cause of the independent variable to the dependent variable. It's very limited in scope. So we just have to be accepting of that and understanding of that when we read results. And then lastly, it's our reliance on significant testing. And this might seem a little bit backwards because that's how we determine the outcomes of any study, right? We run it through a statistical test and we see the likelihood of the relationship that we observed. the likelihood of it occurring. So this is a limitation and an issue, but more than anything, it's just something we have to have an understanding of. It's not that we run an experiment once, find a significant result, and that we found a cause and effect relationship. We really need to understand that we do have probability taking its part here, that we can have errors in our findings, and that we need to be mindful that significance testing, though it's a wonderful way to logically put our questions into statistical practice, it can still be flawed. And that's where our theory and background information and repetition come in to help confirm these findings, right? Helps to confirm our findings. So that's our overview of experimental design, different types of considerations and limitations of experiments. I hope you enjoyed, and I'll see you over on our next lecture.